Hello! Welcome to Squid Essays. Inking your academic pen. My name is Julie and I will be your instructor as we read through the endocrine regulation of sex change phenomenon in marine fish. The article source is embedded in the video description below. Feel free to read along. Please note that you can get a custom, plagiarism-free essay for as little as $10 a page at Squid Essays. The link is in the description. Content 1. Introduction 2. Endocrine disruptors 3. Sex changes related to endocrine function 4. Conclusion 5. Bibliography. Introduction. Nowadays, it is a well-known fact that endocrine systems are found in most varieties of animal life. At the same time, the recent researches have revealed the fact that changes of the endocrine system can have quite unexpected consequences. For instance, some marine fish under the impact of endocrine disruptors and changes of endocrine system can change their sex during the course of their life cycle. Endocrine disruptors. Basically, it is endocrine disruptors that are responsible for such a dramatic change of the organisms of marine fish. It should be pointed out that the disruption of the endocrine system can occur in different ways. In this respect, it is worthy of mention that some chemicals mimic natural hormones, fooling the body in over-responding to the stimulus, or responding at inappropriate time. Other endocrine disruptors block the effects of a hormone from certain receptors by blocking the receptor site of the cell. At the same time, others directly stimulate or inhibit the endocrine system and cause overproduction or underproduction of hormones. In recent years, some scientists have proposed that chemicals may inadvertently be disrupting the endocrine system of human and animals, as well as marine fish. In the case of the latter, there is strong evidence that a chemical exposure has been associated with adverse developmental and reproductive effect on the fish. Sex changes related to endocrine function. The recent researches and experiments reveal the basic sex changes that are related to the functioning and changes of endocrine system. In this respect, it is worthy of mention one of the experiment which aimed at the identification of alterations in gene expression associated with MeHg exposure, specifically those associated with previously observed changes in reproduction and reproductive biomarkers. Fathead minnows were special diets which contained the similar concentration of MeHg in the diets of the wild invertebrates and piscivorous fish. In that research, the commercial macroarray in conjunction with quantitative polymerase chain reaction was used to examine gene expression in fish in relation to exposure to these environmentally relevant doses of MeHg. In the result of the research, it was found out that the expression of genes commonly associated with endocrine disruption was altered with Hg exposure. To put it more precisely, the researchers observed a marked upregulation in vitellogen and mRNA in individual Hg-exposed males and a significant decline of vitellogen gene expression in female fish with increasing Hg concentrations. Other genes identified by the macroarray experiment included those associated with egg fertilization and development, sugar metabolism, apoptosis, and electron transport. The researchers also revealed the fact that there were differences in expression patterns between male and female fish not related to genes specifically associated with reproduction, indicating a potential physiological difference in the reaction of the females and males to MeHg. Conclusion. Thus, taking into account all above mentioned, it is possible to conclude that the endocrine system plays an important role in the functioning of marine fishes and the changes in its functioning can affect dramatically the sex of the fish. The experiments reveal the facts that such changes can occur under the impact of endocrine disruptors which lead to the changes of the functioning of the endocrine system of fish affecting their sex to the extent that it can be changed. In this respect, the experiment with the use of MeHg is particularly noteworthy since it provides practical evidence of such changes. Consequently, it is possible to estimate that the effect of endocrine disruptors on organisms, including marine fish, can lead to serious changes of their functioning. Bibliography. 1. Armstrong, F. Effects of mercury compounds on fish. In Biogeochemistry of Mercury in the Environment, A9AYTZ2H4J, ed. New York, Elsevier North Holland Biomedical Press, 1979, 657-670. 2. Arnold B.S., et al. Effects of methylmercury on plasma estrogen and 11-keto-testosterone in Nile tilapia, Oreochromis niloticus, Abstract. In Abstracts of 19th Annual Meeting of the Society of Environmental Toxicology and Chemistry, 13-19, November 1998. Charlotte, North Carolina, Pensacola, Florida, SETAC Press, 145. 3. Beiswinger, P.J., et al. Glyceraldehyde 3-phosphate dehydrogenase activity as an independent modifier of methylglyoxal levels in diabetes. Biochem Biophys Acta, 2003, 1637-98-106. 4. Bjornberg, K. et al. Methylmercury exposure in Swedish women with high fish consumption. Sci Total Environ, 2005, 341-45-52. 5. Bloom, N.S. on the chemical form of mercury in edible fish and marine invertebrate tissue. Ken J. Fish Aquat Sci, 1992, 49-10-10-1017. 6. Byrne B. M., Gruber M. The Evolution of Egg Yolk Proteins. Prague Biophys Small Bile, 1989, 53, 33-69. 7. Chuang D. M., Hoxie, Synadol VV glyceraldehyde 3-phosphate dehydrogenase, apoptosis, and neurodegenerative diseases. Anu Reverend Pharmacol-Toxical, 2005. 45, 269-290. 8. Colborn, T. Developmental Effects of Endocrine Disrupting Chemicals in Wildlife and Humans. Environ Health Perspect, 1993, 101 378-384. 9. Cole, D.C. et al. Blood mercury levels among Ontario anglers and sport fish eaters. Environ Rees, 2004, 95 305-314. 10. Drevnik, P.E. and Sandheinrich M.B. Effects of Dietary Methylmercury on Reproductive Endocrinology of Fathead Minnows. Environ Sci Technol, 2003, 37 4390-4396. 11. Elliott J. Effects of Estradiol 17? On serum calcium and vitellogenin levels in rainbow trout. J. Endocrinol, 1979, 8354-55. 12. Endocrine Disruptor Screening and Testing Advisory Committee. EDSTEC Final Report. Washington, D.C., U.S. Environmental Protection Agency, 1998. 13. Friedman, A. Low levels of dietary methylmercury inhibit growth and gonadal development in juvenile walleye, Stysostedion vitrium. Aquat Toxical, 1996, 35 265-278. 14. Fine-Eikens, K. An evaluation of methylmercury as an endocrine disruptor in largemouth bass. In 19th Annual Meeting of the Society of Environmental Toxicology and Chemistry, 13th to 19th November 1998, Charlotte, North Carolina, Pensacola, Florida, SETAC Press, 146. 15. Garcia Morales, P. et al. Effect of cadmium on estrogen receptor levels and estrogen-induced responses in human breast cancer cells. J. Biochem, 2004, 269 16896-16901. Thank you for taking your time to listen to me. I hope you enjoyed learning about the endocrine regulation of sex change phenomenon in marine fish as much as I did. Kindly remember that you can get a custom, plagiarism-free essay at Squid Essays for as little as $10. The link is in the description. See you next time.